Welcome to Agronomy 1. This subject is part of the Bachelor of Agriculture and Technology and the Bachelor of Equine Studies. These degrees are offered at Melbourne Polytechnic, with the Bachelor of Agriculture and Technology being a partnership with La Trobe University and Melbourne Polytechnic, while the Bachelor of Equine Studies is a Melbourne Polytechnic degree. To find out more about this course and the other courses that we offer, please visit our website at www.melbournepolytechnic.edu.au. My name is Dr. Nikki Cooley, and I'm going to talk you through Lecture 4 on the topic of crop physiology. In this lecture today we are going to discuss a number of topics. We are going to start with photosynthesis and assimilation. We are going to then look at carbon metabolomic plant classification. We will review plant growth and expand on what we have already covered in this subject. And we will finish with an understanding of phenology based on both a case study that will be presented and some research you will do yourselves. Please note that these are the basics of crop physiology and that they apply to all crops. In fact, they apply to a commercial production of wheat, an establishment of pasture, or even plants in your garden or a plant in the pot on your windowsill. In the subject plant physiology, which we do in Year 2 in the Bachelor of Agricultural and Technology, you will learn a lot more details about these elements that we will discuss today. Please note that you are only learning an overview for this basic level subject. Let us start photosynthesis, also called assimilation. This is really where energy is produced in plants. It's one of a kind. Many of you will have come across photosynthesis at some point. You would have heard about it or you would have learnt it at school. It is when a plant enables the capturing of the energy from the sun and converts it into a chemical reaction. We call the process photosynthesizing. All plants photosynthesize, and in summary, you can think of it as in the equation on the slide, which may look complicated at first, but once broke down is quite simple. Carbon dioxide is a gas found in the atmosphere. Plants are able to what we call fix or capture the carbon dioxide. With the help of water molecules and the assistance of some light and some clever plant enzymes, plant can produce glucose. The C6H12O6 formula is the formula for glucose. The byproducts from this reaction are oxygen and water. You will see that this chemical formula is balanced. What this means is 6 molecules of carbon dioxide plus 12 molecules of water with some light and some help of some clever mechanisms and plant enzymes. 1 molecule of glucose is made, 6 molecules of oxygen are released, and 6 molecules of water are also made. It is very advisable for you to learn every input of this diagram for this subject, as it is the basis of how plants produce energy and therefore grow. It's very significant in the commercialization of the industry. So let us review what is photosynthesis. Photosynthesis is using energy from the sun, e.g. light, to convert the gas carbon dioxide with water into sugar. It releases oxygen gas and the byproduct of water. It is interesting to note that all oxygen gas found in our atmosphere has been released from plants. And it is not an overstatement to say that we would not exist as humans without oxygen in the atmosphere. Let's start at the very basics of this process. The first thing to note is that photosynthesis is enabled in plants because of a clever combination of chemistry and plant structure. So when we are learning about photosynthesis, we need to consider the structure at the same time as we're understanding the process. So, the first component of a plant structure I'd like you to take on board is the chloroplast. The chloroplast is the organelle where both components of photosynthesis occur. You're about to learn that photosynthesis occurs in two stages, stage 1 and stage 2, and both of these occur in the chloroplast. The chloroplast is located in plant cells. Usually, most chloroplasts are located in the leaves, but some can be found in the stem. Now we understand the basics of photosynthesis, let's build on this and explore it in more detail. It is in fact a very complex process, and it involves both chemical reactions and biophysical processes, hence why the structure is important to understand. So we've learned that photosynthesis occurs in the chloroplast, and the next component of photosynthesis we're going to learn is that it occurs in two stages. The first stage is referred to as the light requiring actions, and their name very much suggests their function. It's the bit where the sun or protons are captured. The second stage is the non-light requiring reactions. You can think of this as where all the chemistry occurs. Let us learn about the light requiring reactions, or the first stage of photosynthesis. Well, as the name suggests, light is what is required for this reaction. Light comes from the sun, but it can also come from light bulbs. The sun emits many different wavelengths. There are microwave wavelengths, for example, and there's visible wavelengths. Photosynthesis only uses wavelengths in the visible spectrum. It's called the visible spectrum because we can see it, unlike microwaves which we cannot see. In the visible spectrum there are lots of photons. A photon is a unit of light that plants use in photosynthesis. This is how they require energy. They capture the photons. They do this in the suborganelle of the chloroplast called the thylakoids. So let us review. The thylakoids are the location where plants capture photons from the sun. Photons occur in the visible spectrum, and plants using thylakoids are able to capture the energy from these photons. In order for a plant to optimize or get the most out of photosynthesis, it would ideally follow the sun through the day. So scientists a long time ago asked the question, can plants follow the sun? Well, in a way that humans can follow the sun, plants can't. They can't get up and move. But they do have mechanisms that enable to optimize the sun and capturing of the photons. The chloroplasts, for example, can move very slightly. They can orientate themselves slightly to capture the photons from the sun. Another adaptation that occurs in some species of plants is that the leaves will move. That is, that they can change their angle to enable optimal photon capture. When a plant can move, it is called a tropism. When a plant or a leaf moves and follows the sun, it is called phototropic. If you've ever seen a sunflower follow the sun, this is a great example of phototropism. We have understood the light reaction. It would be good to put some images with the processes I have just described. I'd like you to stop the video here and go to the link on your handouts. This video will give you a visual interpretation of photosynthesis. It will start with the light reaction and then in a logical manner move over to the non-light requiring action, or the part where all the chemistry occurs. I'd like to introduce the non-light requiring action through this video, as I think the illustrations are very good and will enable your understanding. Once you have watched the video, please answer the 7 questions you see on the slide. What is a photon? What proportion of light is used in photosynthesis? What compounds capture the light? Where does glucose synthesis occur? How many photosynthesis systems are there? What is ATPase? And what is the first carbohydrate made by photosynthesis? Have a go at answering these questions and then look at your handout notes. On your handout notes, the answers will be presented and you can compare that with what you thought. If you've answered all the answers correctly, you are now ready to move on to the next part of the lecture. If, however, you made a mistake, can I please recommend you watching the 6-minute video again? It will really aid in your understanding. Most students need to watch this video 2 or 3 times before they have a full understanding of both the light and the non-light requiring reactions. That you have an overview or an overall understanding of photosynthesis, how the plant captures light energy and how it converts it into chemical energy. It is worth us looking at this process from a commercial perspective. So why are we really interested in photosynthesis? In order to answer this question, I just want to define photosynthesis and photosynthetic rates. Photosynthesis is the process, while the photosynthetic rate is how fast that process will occur. From a commercial perspective, we always like to try and obtain high photosynthetic rates. That is, it means that the plant is working really fast, it's capturing lots of light photons and it's making lots and lots of molecules of glucose. So high photosynthetic rates will result in more energy production, which will result in more economic return. Now remember, energy production is in 2 forms. It's in ATP and NADPH, which are compounds of energy, but it's also in the production of the sugar glucose. Glucose can be moved around the plant and it can be made in more complex compounds, which can then be used as energy for growth. In a simple example, high photosynthetic rates, which can be abbreviated to PN, or sometimes you just see this as in a capital A, short for assimilation rate, will equal lots of adenosine triphosphate, ATP, and NADPH, which equals simply lots of plant growth. Although you are beginning to understand there are many components to that process, but the concepts are simple and can be easily followed. For example, if you have a high photosynthetic rate, you will get more pasture. Let us say you may be growing pasture for a horse stud. Therefore, if you can produce conditions that enable high photosynthetic rate, you'll produce more pasture and your horses will have more plants to eat. Or higher photosynthetic rates may equal bigger wheat plants. This will equal more crop, more biomass that you can harvest. Sometimes wheat is grown as animal fodder, but more often it is grown for grain. That grain is sold for either pastas or bread. Therefore, the more energy you can put into producing grains, the more grains you'll produce, and therefore the more money you'll get. Let us look at another industry, tomato plants. Again, the higher the photosynthetic rate, the more glucose you will produce, the more energy or glucose that can then be translocated or move into your tomatoes. The more tomatoes you may have, or your tomatoes may be bigger, depending how your plant is distributing your sugars. So, in essence, the higher the photosynthetic rate, the more money a commercial grower will make. The photosynthetic rate tends to describe how fast a plant is photosynthesizing. But you can also think of the overall photosynthesis in a plant, or what we call the photosynthetic yield. So that is, if you took a theoretical photosynthetic rate and then you timesed it by the area of each leaf, the more leaves that you would have, the larger the area, the more photosynthesis. So from a commercial perspective, it is not only important to consider the photosynthetic rate of each leaf, but also to consider the entire photosynthetic output or yield. It is also interesting to note that not all leaves have the same photosynthetic rate. For example, a leaf in the sun will usually have a higher photosynthetic rate than leaves in the shade. A very old leaf will usually have a lower photosynthetic rate than a leaf that has just reached full expansion. So now we've understood about the commercial properties of photosynthetic rate and total plant photosynthetic yield. There are other components of photosynthesis that we need to consider. Photosynthesis can have what we call different types of photosynthetic metabolism. Basically, that means that the chemistry part of photosynthesis can change, and in fact they can be divided into three groups. There are what we call the C3, the C4 and the CAM groups, and they all have slightly different photosynthetic pathways. In the example that we used in the video, we learnt about the Calvin cycle, and this nicely describes C3 plants. A lot of plants grown commercially, are C3, but not all. So all of these plants will capture light energy and convert it into chemical energy. It is just the chemical component, or the part that they do slightly differently. C3 plants are called such because their first output in the Calvin cycle is a 3-carbon compound. These two 3-carbon compounds join together to make glucose. C4 plants have evolved a slightly different output. They have what we call a 4-carbon compound output. While CAM plants undergo a different photosynthetic pathway. That is, they store carbon dioxide at night and only do the light requiring reaction of photosynthesis during the day. To put simply, they compartmentalize photosynthesis, and they do this to save water and energy, and usually the CAM plants are quite extreme. For example, cactuses are CAM plants. I don't often farm CAM plants, so I'm going to concentrate on C3 and C4, and I hope in the following example you'll begin to understand some of the differences. I'm going to use the C3 and the C4 grasses as example, because these are often commercially farmed in either as a crop themselves or in complex pastures. So let us look. The initial molecule formed during photosynthesis in a C3 plant is a 3-carbon compound, in a C4 plant is a 4-carbon compound. The growing period of these plants is different. C3 plants tend to have cool season or year-long growth cycles, while C4 plants tend to grow in warm seasons. C3 plants tend to do well in lower temperatures, while C4 plants tend to do better at higher temperatures. C3 plants tend to do well when the moisture or water levels are high, whereas C4 plants have evolved to be good in drought conditions or where moisture is low. C3 plants tend not to need very much light to be efficient, so we say their light requirements are low, while C4 plants require high light conditions. C3 plants tend to be do not have a sensitivity to frost, while C4 plants tend to have frost sensitivity. C3 plants tend to have much lower productions, while C4 plants have higher productions. Most importantly, from a pasture perspective, C3 grasses tend to have high feed quality, while C4 plants tend to have lower seed quality. There are many complex reasons why these features are found and are different in C3 and C4 plants. We will not cover them here, but those of you that go on to do the plant physiology subject will learn about these in more detail. On the slide you can see some pictures of examples of C3 plants and C4. C3 plants include wheat, subclover, lucerne, eucalyptus, acacia and cocksfoot, while C4 plants include saltbush, maize, sorghum, sugarcane, millet, kangaroo grass, buffalo grass and green panic grass. So now we have learnt about the different photosynthetic pathways C3, C4 and CAM, and we have learnt about the basics of photosynthesis. There is a light requiring reaction and a non-light requiring action where the chemistry occurs. We have a good understanding, or at least good enough for this subject, to learn next about plant growth. Photosynthesis enables plant growth. That is, the energy from plant growth, ATP and NADPH, can be used to power photosynthesis. Plant growth is defined as an irreversible increase in dry matter. We explored this extensively in lectures 1 and 2, and we will expand here. Let us look at what we call constituent growth in plants. It is broken into a number of components. We have cell division. Cell division is an increase in number. But please remember that a cell divides first. It splits into two. Then you get cell enlargement. This is where the cell increases in size. It is important to remember that growth requires protein synthesis and that the process is irreversible. It can be easy to measure what you think is a change in plant growth, but it actually just be a change in the amount of water that the plant has at any one time. The third process of growth is differentiation. This is when the plant specializes or becomes the cell it intends to be. Photosynthesis and energy are required at these stages. It is therefore measured by a variety of parameters, some of which are increase in fresh weight, some dry weight, some length, some area, some volume, and some cell number. You have learnt about some of these in your DIY practical, and we will continue to learn about these throughout the course. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase by size up to 350,000 times. That's a lot. In the former, growth is expressed in increase in cell number, and in the latter, growth is expressed as an increase in cell number. While the growth of a pollen tube is measured in terms of its length, an increase in the surface area denotes the growth. The period of growth is generally divided into three phases, mainly meristematic, elongation and maturation, as we have discussed previously. We have vegetative division, which is described by mitosis, and reproduction cell division, which is described as meiosis. If you are struggling to remember what mitosis and meiosis is and what it looks like, please refer to the Living Systems 1 for your notes. This will give you a thorough interpretation, and you will also remember the practical where you saw these processes. It is also worth noting that plants have the capacity for what we call unlimited growth throughout their lifespan. This is important from a commercial perspective, as it can change the way you farm. Therefore, growth can be divided into 3 phases: meristematic, elongation, and maturation. Meristems are the physical regions of where growth occurs. It is important to understand where the meristemic regions are found at both the end of stems and at root tips. If some of your management strategies are cutting off your meristems, then you are likely to stop growth altogether. Meristemic areas are the location of cell division. Growth at a cellular level is principally a consequence of an increase in the amount of protoplasm. There are 2 types of growth. One is called arithmetic and the other is called geometric growth. The differences between these growth types is how the expansion occurs, and the diagram on your slide goes some way to explain this. During arithmetic growth, which is shown on the left-hand side, 1 cell is added at each cycle. So for example, in the first cycle you have 2 cells, in the second cycle you'll have 3, and in the third cycle you'll have 4, etc. Whereas geometric growth types results in every cell dividing, and therefore you end up with many more cells. So in the first cycle you have 2 cells, in the second cycle you have 4, in the third cycle you will have 8. So let us compare the 5th cycle of each process. In arithmetic cell division you have 5 cells, whereas in geometric cell division you have 32 cells, a lot more. Growth is therefore measured by a variety of parameters, some of which can be increase in fresh weight or dry weight or length or area or volume. We can give these types of growth formulas. On the slide you will see the formula for arithmetic growth as well as geometric. The units are given descriptors. That is, LT stands for the length in time. I do not expect you to remember these formulas, but I do expect you to be aware that there are different types of growth, and this can impact on your commercial production. The following slide is a summary of all the different main types of plant growth measurement: fresh weight, dry weight, length, area, volume, and cell number. In your DIY pracs you have started on length. The most common pattern of growth is called sigmoid growth, and it is demonstrated by the simple illustration on the left-hand side. You have dry matter compared with time. In the first part of growth you have what is called a lag phase. This is followed by a log phase. This is where growth occurs very quickly. Then in section C you'll have a decreasing rate. In section D you'll have a steady rate, and then the last section E you'll see a decline, and this is usually due to senescence. Senescence is the technical word we use for plant reduction or cell death. There are many factors which can change the shape and the relationship of plant growth. You have external factors such as the climate, light, temperature or water, day length or wind. Then you have emphatic factors such as the soil, for example the soil texture or the structure, or the OM or CEC. We will learn about these in the soil lecture later on in this subject. Competition for resources from weeds or from insects or diseases or nematodes or soil microbes can all impact on growth. The ability for your plant to respond from a genetic perspective can also be considered when growing plants commercially. That is, how resistant is your cultivar to certain stresses? How good is your photosynthetic rate of your cultivar or the photosynthetic yield of the whole plant? The opposite to photosynthesis is respiration, and we are not going to learn about much that here. But respiration can take energy from the plant. So a plant that has a high respiration rate can reduce growth. The last thing to consider that can impact on growth is the partitioning of assimilates and nitrogen. Assimilates are sugars, for example, how glucose or sucrose or fructose is transported and where it's put around the plant can all impact on your plant's growth. The partitioning of nitrogen is also important in plant growth relations. So, in summary, when we have plant growth, we have three stages: cell division, cell elongation, and cell differentiation. These can all differ across cultivars, and there can be different types of cell division: geometric or arithmetic. As well as whole plant growth, there are different stages of plant development, and we call this phenology. In different phenological stages, plant growth can change. That is, some phenological stages can promote high plant growth, while others promote slow growth rate. So, is the rate of growth important? Well, the simple answer to that is often very important, particularly when you're growing for a specific component, i.e. you want both the weight of a grain and good protein in a grain, or you want the yield of a grass with good protein in it. One question to think of is, do growth and developmental stages proceed at fixed rates in plants? Well, the simple answer is no. The climate and environmental factors and cultural practices will often modify these, and in here lies much of the challenge of growing plants commercially. I'm now going to teach you about what we call organ differentiation, or the various stages of plant development. Different plants develop in different ways. Therefore, I am going to start by using the example of wheat development. I will use that as a case study to take you through the various stages. I also want you to research your theoretical crop, that is the one that you have selected for your plant management plan as part of the assessment for AGR1, AGR. So, in essence, we have a number of stages. The following stages that are distinguished in wheat are germination, emergence, tillering, floral initiation, terminal spikelet, first node or beginning of stem, boot, spike emergence, anthesis, and maturity. In many plants that start from seed, you have the process of germination. This is where the seed coat is broken and the new plant comes out. When the coleoptile, which is the structure that comes out of the seed, comes out further, we then describe this as emergence. In your DIY practicals you learnt about germination, and you've also learnt how to measure early seedling growth in this emergence stage, so you'll be very familiar with these stages. There is a whole suite of growth, or what we call tillering of a wheat. The tillering reflects the morphology of the wheat. Floral initiation is the start of the flowering process, and the terminal spikelets is a part of the physical formation that is important in our definitions for specifically wheat. Then you have first node or beginning of the stem. This is then followed by boot, spike emergence, anthesis, which is flowering, and maturity, which is the process where the plant has produced its seed. If we look at the image on the slide, this will help us describe the various stages or phenology of wheat development. You can see at the beginning on the left hand side of the image we have sowing, and there is no plant present here as it's just a seed. Then from sowing we have the process of germination. Once the seed coat has broken, the plant has emerged, we have the process of emergence, which is described by the letter E and is the first physical representation of the plant on the slide. Then you have a number of stages of growth. These can be ranked from double ridge appearance, terminal spikelet initiation, heading, anthesis, beginning of grain, the filling of the grain, and physiological maturity. Once you understand what a wheat plant looks like, you will be able to differentiate these stages. If we make the statement that crop phenology is important in commercial production, in order for us to optimally manage that, we need to be able to define and clearly represent various stages. This is different for all different crops. So for example, wheat, we have emergence E, growth stage 1 GS1, growth stage 2 GS2, and growth stage 3 GS3. These are shown very simply on the slide, and referring to the previous slide, you can see where each of these regions fall into. It is important to understand that different phenological stages are more or less sensitive to impacts such as climate or day length or sowing seed. Therefore, when you are beginning to understand or learn about crop and the growing of crops, it is important that you understand that not only do you have to think about how the whole plant reacts to a certain temperature, day length or sowing date, but actually it's the individual stages that can very much influence the whole plant response. Therefore, learning to categorize and understanding environmental impacts on these stages is important in your commercial enterprise. Each cultivar is different, therefore we learn one species. The table on the slide was produced by the FAO and shows typical time-lapse values for the various stages in spring and winter type germination sown in May at 34 degrees south based on US growing conditions. Various environmental stresses, particularly heat, but also water and salinity, may shorten the wheat growing phases. This is one example of understanding development stage and how phenological maturity may be impacted. Let us look at these from a growing crop perspective. What does it all mean? Well, let us say we're growing a crop like wheat from seed, and we need to consider the sowing time. The sowing time is important as it impacts on the timing of the plant and the climate interactions at all stages of phenology. So, in our example, if a crop is usually sown in the winter, if you sow the seeds too early, you may miss out on rain, leading to reduced yield. If you sow your winter crop in Australia too late, you may get adequate rain, but the temperatures are too low for optimal germination. Frost may also kill or denature delicate seedlings. This same frost may not impact more mature plants if you had your timing correct. Another take on this is if a winter crop is planted too early, optimal temperatures for flowering may not be received. Therefore, your crop could be reduced. If a winter crop such as wheat is planted too late in Australia, the temperatures may be too high for flowering and thus reduce flower number and the development of fruit or grains. On your handouts, please refer to the article abstract "Simulated Wheat Phenology is Improved when Local Temperature is Used." A link can take you to this resource. This link will give you an understanding of ambient or local temperatures and how it can assist in you planning your timing of your crop. Another important stage in crop development for some crops is that of pollination. Some crops require help in their pollination. Crops that do not require help are called self-pollinators. However, crops that require help usually require what we call vectors. An example of a vector can be a bee, or it can be an animal or a bird. In agriculture in Australia, bees tend to be our most important vectors. I would like you to follow the link, which is also shown in your handouts, and this is a very simple description of the importance of bees. After you have read this link about the importance of bees, please have a look at the Wikipedia link. This is a very interesting list of all the crop plants that require pollination by bees. You will see many of these are important commercially in agronomy and also important commercially in Australia. With this lecture, I would like you to research some more information on your theoretical crop for your plant management assessment. That is, in your plant management assessment, each of you were assigned a different theoretical crop, like a grapevine or a grass. Please research the crop phenology specific for your crop, from seed through to the commercial product. If your crop's phenology does not start at seed, but starts as a mature perennial plant, then please follow the defined crop phenology that is important for the commercial product of your plant. For my flexible delivery students, please email me your research once you have completed this lecture and your research. For my face-to-face cohort, please bring to the next class after you have watched this lecture and had some time to research your crops. We will review your phenology for each student. Please use images and descriptions in your descriptions. So that brings us to the end of this lecture. So let us summarize what we have learnt or taken away from this lecture and its associated readings. I hope you now have a basic understanding of photosynthesis, that it requires 2 stages of reaction, that the product of photosynthesis is very important from a commercial perspective, that photosynthesis enables growth and development of crop plants, and that you now have an understanding of what growth is from both this lecture and previous lectures, and also what development and how phenological components can be defined. Please ensure that when learning aspects of this lecture, you consider both your theoretical crop and your wheat as a case study when reviewing specifically the phenological measurements. That brings us to the end of this lecture.